Good afternoon, everybody, and welcome to this EFSA's webinar on protein safety in GMOs. My name is Renilde Schoonjans, and I am the team leader of the GMO Food and Feed Safety Assessment team. I will be your moderator for today. And thank you for joining us and showing such great interest in our work. We have received 600 registrations representing food industry, NGOs, universities, international organizations, and national competent authorities from Europe and beyond. Together with me, we have a team of EFSA colleagues. Our two expert presenters on the topic are Antonio Fernandez, who is Scientific Officer in the Nutrition and Food Innovation Unit at EFSA, and Javier Moreno, who is Chair of the GMO Food and Feed Working Group of external experts, working with us for EFSA. And as Q&A manager, we have with us Telemakos Gumperis, who will be helping me with handling the questions that you can submit throughout the webinar on the Q&A tool that you see on the right-hand side of the presentation window. Let me explain a bit on the webinar structure. Antonio will start by introducing you in his presentation of about 15 minutes to the reasons and the objectives of our activity on protein safety assessment in GMOs. And then Javier will continue in his presentation to explain in more detail the survey that we will launch right after this webinar. This survey is meant to collect inputs from you on this topic. Those inputs can then be used to inform the development of the scientific opinion by the GMO panel. And this survey will remain open until the 18th of February 2024 to allow as many responses as possible from the stakeholders. And then follows our live Q&A session, which will last for about 20 minutes, and we will be using the questions that you have been posting. Some housekeeping rules that you will see here on slide is that you are automatically connected in the listening mode only. And questions should be submitted in English via the Q&A chat, again on the right-hand side of your presentation window. You may post them throughout the entire event, and Telemakos and I will try to aggregate them and pass them to the respective speaker to be addressed during the Q&A after their presentations. We like to ask you to keep the questions as short as possible because we want to address as many as possible during that Q&A session. What is out of scope here are clarifications about the authorization process or any other legal aspect. We keep them out of scope of this webinar and therefore will not be addressed. In general, if we do not manage to address some of your questions and they remain unanswered due to time limits, you can resubmit them to the Ask a Question Connect. This is an EFSA tool that you find by following this link on screen. So this event will also be recorded for future reference, and we will publish it together with the presentation on EFSA's website. I do hope you enjoy this webinar that we have prepared together, and I now pass the floor to our first speaker, Antonio Fernandez, who is Scientific Officer of the NIF unit.
Thank you, Renilde, and welcome to everyone to this webinar. On the screen, as described by Renilde, you have the agenda of today, and I will take care of the first part, the self-task Monday. I will present it to you, and afterwards we'll give the floor to Javier for the open survey, and we go into the questions. We have structured this presentation to answer three main questions according to us is in the head of everyone. That is, why are we doing this activity? What we try to achieve, and what is needed from stakeholders. So in relation to the first question, why are we doing this activity? I would like to start from the overall umbrella, the risk assessment of GMOs, worldwide speaking, is performed following the principles set by Codex Alimentarius already in 2003. These principles borrow what at the time was the safety assessment doing for chemical risk assessment, but we know that large molecules, proteins, are different than small molecules, and several in the scientific community raise the need to improve and modernize protein safety assessments. As mentioned, we have to realize that Codex was set 20 years ago. If we move to the next slide, thank you. To zoom in and to provide more context on what is protein safety, what are we after? It's protein toxicity and allergenicity in GMO plants. And to zoom in into the context of Europe on the experience we have gained over this year, as mentioned, Codex Alimentarius principles has been embedded into Regulation 543 and EFSA guidances, and the main information considered relates to knowledge on the source of the protein itself, a history of safe use, bioinformatic analysis, in vitro studies, and in vivo studies. This information is considered on a case-by-case approach, as I will detail in the next slide. Regulation 543, the present requirements, we have depicted those here to you, mainly for toxicity and allergenicity. In the case of toxicity assessment of newly expressed proteins, the case-by-case approach is the following. If history of safe consumption is duly documented, specific toxicity studies are not needed. While where specific testing is required, the applicant shall provide molecular and biochemical characterization of the newly expressed proteins, bioinformatics searching for homology to proteins known to cause adverse effects. Here we are talking about the primary sequence, stability of the protein, for example, influences of temperature and pH, degradation of the newly expressed proteins to proteolytic enzymes, and here there is a direct mention into the classical pepsin resistance test, and finally 28-day toxicity study. Depending on the outcome of this information, additional investigations might be needed. While in the case of allergenicity assessment of newly expressed proteins, also following similarly a case-by-case approach, it should contain bioinformatics searching for homology with known allergens, also based on primary sequence, a specific serum screening, and this is for cases where there is either a sequence homology or a structure similarity to a known allergen, or when the source of the gene is considered allergenic. Pepsin resistant test and in vitro digestibility test, and additional studies, cell-based animal studies if needed. So overall, throughout these years, this has been noted as an effective way of assessing GM plants, but the panel has also commented on the experience gain and on different elements that would add to improve this safety assessment. And for example, we included here the need for specific definitions on some key elements, such as what is meant by similarity and how similar is similar, what is meant by history of safe use and what criteria could be included to move forward this field, and additionally, what is meant by thresholds. Can we include exposure also in the safety assessment, and specifically, for example, in silico tools, what type of thresholds can be included? In addition to this need for definitions, the GMO panel has also experienced some challenges in some specific cases for the assessment of newly expressed proteins when these are difficult to extract and purified. We have some examples, membrane-bound proteins. We also have reflected on those cases where GMOs contain a high number of newly expressed proteins. And finally, in those cases where partial similarities to toxins and allergens are identified. These are challenges experienced by the GMO panel. And finally, related to the methodology, the GMO panel has reflected on opportunities, on the need for alternative methods, the need for an experimental shift within the weight of evidence approach, and opportunities for standardization and validation of new methods. So in essence, the EFSA GMO panel has decided to embark in this activity. At the bottom of this slide, you have the link to the mandate. And basically, we have summarized here the information you will find in the mandate. To start with, what is the current situation, identifying the different requirements I just explained in my previous slides for toxicity and allergenicity assessment, with the history of safe use as a specific element at the beginning of this evaluation. Which challenges are we facing, with some very clear examples, as it is in the case proteins are difficult to characterize and test. We have examples of membrane-bound proteins, transcription factors, and last, we also reflected in the mandate on potential future opportunities to improve current practice and provide complementary alternative methods. This based on the experience gained and new protein safety assessment methodologies that are today available and were not there 20 years ago. The next question I mentioned at the beginning of my presentation, what we try to achieve. In practical terms, we will produce a scientific opinion reflecting on current practices, challenges, and future opportunities of protein safety in GMOs. And specifically, the terms of reference has been listed here. Terms of reference 1, lessons learned from experiences in the assessment of newly expressed proteins in the last 20 years, including more recent complex cases I explained before. Terms of reference 2, building on the experience and issues identified, develop a critical appraisal of new methodologies available with the potential to be used as complementary alternative testing strategies to current methodologies. Terms of reference 3, roadmap for future implementation of such complementary alternative methods in risk assessment strategies. And finally, terms of reference 4, recommendations for further research to address methodological development needs. Yeah, in my last slide, we wanted to show to you, this is a screenshot of a poster that we presented at a Eurotox this year, and we wanted to show some examples of what elements will be discussed by the GMO panel and in the opinion. And in the weight of evidence approach, the elements that we are at the moment discussing are, for example, issues such as history of safe use, as I explained at the beginning, protein characterization, mode of action, stability, source organism, phylogeny, structural functional similarity to known safe proteins, similarity to known toxins and allergens, fate in the gastrointestinal tract of the proteins, interactions between proteins, and other elements that the GMO panel considers will add to the weight of evidence approach. The way these elements are to be embedded in this weight of evidence is at the moment under discussion, and the idea is to describe a tier approach where, for example, for toxicological assessment, the use of in vivo studies are to be pursued only if concerns are identified. On the need, for example, to develop more fit-for-purpose databases, like, for example, in the allergenicity assessment and the ranking of allergens. And the tools we have been considered for this activity goes into the new philosophy for the new approach methodologies. For example, in silico tools, information derived from a structure of the novel proteins, structural similarities, or for example, in vitro testing, information gathered from stability of the newly expressed protein during processing, storage, or gastrointestinal digestion. So again, these are examples that were provided. And I will move into the next slide. Yeah, I will pass the floor to Javier Moreno, chair of the Food Feed Working Group, that will now tackle the next question, what is needed from the stakeholders and the survey purpose. Thank you very much.
Thank you, Antonio. Hi, everyone. Just now it's time to focus on the survey and to give you some indications about the meaning of the questions that you will face when you complete this survey. In this slide, you can see practical information about the survey launched by EFSA, which will be open from today until the 18th of February next year. Here you have also got the QR code for the survey link in this slide. And the main purpose of the survey is to capture feedback from stakeholders to better understand potential alternatives based on current knowledge and recent development that could be incorporated in current and risk assessment in the future. Your feedback will be also important to prioritize research needs and to help to define a roadmap for action. I will say that this survey is a key step in the mandate on protein safety assessment, and the aim is to gather as much information as possible from you about how to proceed in the future. The survey answers will be kept as long as needed for the purpose of the survey analysis, and of course the data will not be used for any other purpose. So please don't be shy and feel free to share with EFSA any views you may consider useful for the context of this mandate. Next slide, please. Yes, the survey comprises a series of general questions based on the terms of reference that Antonio presented before. In addition, you also will find different sub-questions to give the opportunity to provide more focused information because the main questions are more general according to the terms of reference. So as you can see here, the first question of the survey is which is the common strategy for the assessment of the new expressed proteins in GM food and feed. The purpose of this question is to do, let's say, a mapping work about the outcome of the most common risk assessment strategy applied for protein safety assessment so far. As Antonio mentioned before, this strategy was developed by the Codex Alimentarius and then adopted by competent authorities. And this strategy has efficiently worked since its implementation around 20 years ago. But as science is continuously evolving, we are receiving new applications with different approaches. We think that it's now time to recap about this type of outcomes. Concerning the sub-questions, the survey includes the following, as you can see here: in how many cases the current methods have identified issues or safety concerns for human or animal health, what type of animal studies is requested by the different risk assessment authorities and under which circumstances, or which studies and methodologies have been more or less meaningful to identify potentially adverse effects. Basically, these first questions aim at taking advantage of the experience gained in the past. When developing new approaches to be potentially incorporated into risk assessment strategies, it is essential to assess past experiences and determine how new technologies and methodologies can potentially replace or complement some of the data requirements which are currently in place. Next slide, please. There is a second general question belonging to the first terms of reference. This is which are the complex cases requiring a different approach for their assessment. Antonio already mentioned some of them. Some of these examples have been already identified by the GMO panel, and indeed have been previously highlighted in different scientific opinions endorsed by the panel, as you can see in the right side of the screen. Some of these cases we deal with in the scientific opinion dealing with the risk assessment of synthetic biology-derived plants, or the document describing development needs for the allergenicity and protein safety assessment on food and feed products derived from biotechnology. Some of these examples are in the left side of the screen. For instance, how to deal with GM plant expressing a high number of protein to test. As you know, Codex approach was developed by targeting mainly the assessment of a few newly expressed proteins in each GMO. Another example is how to assess proteins which are difficult to extract and purify, like the membrane-bound proteins. In these particular cases, there can be limitations in the production of enough text material of sufficient quality for safety studies. Also, if it is possible to better understand the biological relevance of partial similarity of newly expressed proteins to toxins or allergens, and etc., or any other cases that you may have in mind. Next slide, please. Once evidence is on gain experience is collected, the purpose is to move on the second term of the reference, which is mainly based on the critical appraisal of new methodologies available with the potential to be used as complementary or alternative testing strategies to current methodologies described in legal frameworks. As indicated before, the Codex Alimentarius and guidance documents from relevant competent authorities have successfully addressed protein safety assessment of GM applications. However, new developments in the field may call for a revision of some elements of the risk assessment in some particular cases. So within this term for reference, there are two main questions. The first one is which complementary alternative methodologies can be used for protein safety assessment, considering hazard identification and hazard characterization. Some examples for further thinking are in the graphic display in the right side of the slide. For instance, the full understanding of a protein's mechanism of action, especially those leading to potential adverse effects, can be a critical piece of information for risk assessment. New knowledge gained from concepts like structural phylogenetics can be also very helpful. For instance, the investigation on how protein structures are polished by the process of evolution and how they are conserved and interrelated could be useful information to be incorporated in risk assessment, and this can be done from the combined use of evolutionary and structural information for proteins. Also, the investigation of the potential similarity of newly expressed protein to not only toxic or allergenic proteins, but also to proteins considered safe could be also an important piece of information. In this context, for sequence homology comparison, alignment-based methods like FASTA and BLAST are normally used, but they can be insufficient in some cases when the assessment of functional similarity is needed. For instance, for most of the proteins, the toxicity is determined by their function rather than by their primary amino acid sequence. So in such cases, the search for functional similarity of proteins, which is related to the structural similarity, could be needed. In these particular cases, until a few years ago, the incorporation of a structural alignment comparison for proteins were not very feasible in risk assessment because not so many three-dimensional structures were experimentally elucidated, and the tools to perform in silico prediction of protein modeling were not, let's say, highly reliable. However, if we consider the significant advances achieved by recently developed computational tools for predicting three-dimensional protein structure, the use of structural alignment studies identifying similarities in shape and three-dimensional rearrangement between proteins could play a key role in protein safety assessment in the close future. Also, a better understanding about the gastrointestinal fate of proteins during digestion could help to make a more robust and scientifically sound assessment. However, due to the novelty of some of these in silico and in vitro tools and their lack of harmonization and standardization, including their readouts, it is currently difficult to evaluate the relevance of this information in a risk assessment context. In general, there is a lack of guidance on how to interpret the outcome and how to embed it in a weight of evidence approach. These are the main reasons behind the second general question within this term of reference that you can see at the bottom of the slide, and which reads as follows: what stepwise approach following a weight of evidence should be used for the safety assessment of newly expressed proteins in GMOs? Next slide, please. The key element for the for the third term of reference is the proposal of a roadmap for future implementation of such complementary alternative methods in risk assessment strategies. New approach methodologies, the so-called NAMs, include novel in silico and in vitro tools already used in a common way in research, and some of them could be integrated in the protein safety assessment. However, as they are not routinely used in risk assessment, a roadmap for action could be needed to pave the way for their practical implementation and to be worldwide accepted by risk assessors and stakeholders. Once again, the feedback received from the survey could really help on how thinking ahead and how to propose practical solutions to move the field forward. Using a similar philosophy, the GMO panel recently proposed a roadmap to improve the weight of evidence approach used for allergenicity risk assessment, as you can see in the bottom right of the screen, which, among other aspects, define knowledge gaps on allergenicity prediction, identified specific research needs, determine how new basic research findings and technological development can improve the current risk assessment methodology, and also prioritize basic research funding. So the two questions included in the survey belonging to the roadmap are how these new methodologies can be introduced as complementary alternative testing strategies in the overall weight of evidence approach for protein safety, and how should the outcome of these new methodologies be interpreted to inform the overall weight of evidence approach for protein safety. As mentioned before, the aim of these questions is to allow a practical implementation of some specific tools or approaches that could be identified as useful and informative for the protein safety assessment. Next slide, please. In addition, this roadmap could also serve to identify key knowledge and methodology gaps and research recommendations to provide information about how to fill these gaps. So the last term of reference is dedicated to these issues, and there are 2 questions about gaps and research activities. Also, there is a third question about what factors could be considered for building trust and confidence in these new methodologies. Some initiatives in this direction could be the inclusion of diverse perspective and stakeholder engagement, perhaps the proposal of proof of concept projects to demonstrate the advantage of these new methodologies. Also, maybe it's important to produce transparent communication of methodologies and findings, or regular updates and additional opportunities for exchanges. Finally, I also like call your attention about the last item of the survey, which is basically a catch-all comment box where you could include any additional comment or aspect that you may miss along the survey. Next slide, please. So before finishing, let me remind you the next steps of the mandate. The survey will be open from today till the 18th of February. A public consultation on the draft opinion delivered by the GMO panel is expected to be carried out from the end of June next year until mid-September. The adoption of the scientific opinion by the GMO panel is planned by the end of next year. In addition to the survey and public consultations, information we are gathering via EFSA developmental projects will be considered as possible. And hopefully the publication of the scientific opinion will not be the end of the story, but the beginning to find a science-based, workable, internationally agreed, and fit-for-purpose approach for the protein safety assessment. Thank you very much for your attention, and now I give back the floor to Renilde. Thank you.
Yeah, thank you, speakers. It was a detailed presentation to explain to us what we have learned in the couple of cases that came recently that are just a little bit different because they are innovative products. They have different type of genetic modification for a very particular trait that is maybe good for the consumers. But it's our job to ensure the highest standards of consumer protection. So we are here to question, is there something we can do more or differently? Also for these most new and more complicated cases. So thank you very much. To start our Q&A, I would like to first look at, so there are a couple of very good questions already lined up for our speakers coming from the Q&A tool that you can still keep populating. But to start, I want to look back to the question that EFSA posed to you during the registration, because 27 respondents already provided us with inputs on new risk assessment methodologies. For instance, low dietary exposure levels as integral parts of safety assessment, strengthened in silico and in vitro methods to understand functional properties and mechanism of action of the protein, or extend bioinformatics beyond sequence alignments and include, for instance, amino acid distribution. Another idea is try to extend the digestibility analysis of unknown proteins or for unknown proteins. And another one is to look at in vitro gastrointestinal toxicity assays. Antonio, I would like to pass the floor to you and ask you to give your first impressions on these inputs and their feasibility and usefulness. Thank you.
Thank you, Renilde, and thank you again to all participants for attending today and also for providing already quite good indications on the way we should go. Just to give you some indications on the feedback we have received so far, gives into the direction of highlighting. So for the EFSA GMO panel to highlight, to better understand what current methodologies are obsolete or in need of revision, what new methodologies can be used as alternative methods as defined by Renilda in some of the comments from you, to define criteria on how this can be done within the weight of evidence approach, including how to interpret different outcomes of the new methodologies. And also last but not least, some of you also commented on the need that several of these improvements, changes will require international agreement and consensus. So we also understand all this is challenging and will require time, but also to get it done, there is a need to start, and this is basically what the GMO panel is trying to achieve, to give it a start. And within a year, how far we can go, it will depend on how much feedback we get from you. So the message also, as reiterated by Renilda, we want to give to you and by Javier is, please, in the survey, be as detailed as possible so that we can make use of it in the scientific opinion. To give some time to you, Javier, I will address maybe a couple of questions we have already received. The first one is, it reads, toxicity assessment requires homology comparison with proteins known to cause adverse effects or allergenic proteins. Do lists or databases of toxic and all allergenic proteins exist that shall be used for comparison? This has been the question from the audience. So far, according also to Codex and the current requirements, we are following the main focus of the bioinformatic analysis in this case is based on the primary amino acid sequence. As mentioned by Javier, also a new knowledge has appeared on structural similarity, for example, and these are aspects that the GMO panel is also exploring how to use those elements. For the databases, also from this side, there are databases that are already in use for risk assessment. And for sure, some ideas will be for future improvements to have more fit for purpose risk assessments. And just to give you also some indications from some participants that already address some of these elements in relation to the bioinformatics-based approaches. Some considerations from the participants is at the moment, the way they are framed would yield too many false positives in case multiple proteins are mutated or introduced in parallel, and this will require complementary follow-up assessments. So these are elements we will discuss and reflect in the scientific opinion. In relation to the next question, when toxicity testing is required, no genotoxicity has been reported. Is there a need of genotoxicity test as needed for a novel food, for example? So the way safety assessment of proteins are done in GMOs are based on a weight of evidence approach. And if based on this weight of evidence indications of toxicity, for example, are identified, further assessment, targeted assessment are pursued. Also in this case, I will read some comments we received from the feedback already received. The requirement expectations for in vivo toxicity studies does not align with the recognized need to reduce animal usage, and new approach methodologies, including in vitro studies for assessing proteins, are needed. So these are some also feedback we received from you. And again, in the survey, please be as specific as possible. Javier, I give the floor to you for the next questions.
Thank you, Antonio. Yes, one of the questions is related to protein stability. Indeed, the question is if can we provide a view on how stability of the protein is characterized in a quantitative way? Okay, we normally check the protein stability regarding the effect of pH and temperature on mainly enzymes. So in that particular case, we assess how this range of pH and temperature could affect the in vitro enzymatic activity. And we also assess the protein stability regarding the resistance to in vitro digestion, mainly pepsin, although we also received some studies dealing with a sequential digestion of pepsin and pancreatin. Okay, in the case of the digestibility, normally we assess studies dealing with the detection of the monitoring of the digestion process with SDS page, with protein staining or Western blotting. So normally we assess the different bands just appearing or disappearing during the digestion process. Indeed, one of the aspects that we would like to get into more details is to be able to somehow reach a consensus about some criteria to help to interpret the outcome of these in vitro digestion studies. For instance, we are thinking we are discussing about how to find potential criteria to identify digestion fragments as relevant for risk assessment of sensitization or elicitation allergenicity, meaning by that abundance of the fragment, the persistence, I mean, how long is resistant during the digestion process, also the molecular size, and maybe others. Some of these aspects were already highlighted in allergenicity guidance published by endorsed by the GMO panel in 2017. Also, we are thinking that it's maybe important to reach a consensus or definition about the criteria for classifying a protein as resistant or liable to digestion, as well as the risk implication of such data. So I think all of these criteria could help to quantify the protein stability, at least derived from the in vitro digestion studies. Okay, I have another question. Okay, another question is, in respect of mode of action, don't you envisage a routine consultation of gene ontology tool as part of the process? Yes, indeed, we are already moving in that direction with internal discussion. For sure, we think that the gene ontology analysis is very important to identify in general biological processes, but also molecular functions, which are key in this case of mode of action. So yes, the answer is we are consider this analysis. So I'm back to you, Antonio.
Thank you, Javier. Yeah, thank you, Javier. I will tackle another two questions. One, it reads, it is an already known allergenic or toxic protein produced in a GMO organism considered under GMO legislation. Here, as described during the presentation, the safety assessment of GMOs is performed following a weight of evidence approach. And obviously only those with no allergenic or toxic properties are considered safe and have a positive outcome. So from this side, the scientific opinions are published in the EFSA website. And I would say that we provide quite many details on the way the safety assessment is performed. So this, you can identify those in the website for the scientific opinions. In the next question, would you provide examples of a newly expressed protein that was difficult to characterize, as well as new testing methodology that should be considered for inclusion in safety assessments? We have some examples of NEPs that are difficult to characterize. For example, membrane-bound proteins, or for example, transcription factors. These are proteins that in many cases are expressed at very low levels, sometimes below the limit of detection or quantification, that make it difficult its tests. And this is definitely an aspect that, as mentioned, the GMO panel has been reflecting, and well, is one of the reasons of why we are embarking into this activity, to define new elements or alternative elements to assess this type of proteins where we have problems to apply the general way of weight of evidence as in regulation or Codex Alimentarius. And the next question asks, could you please give an example where additional studies, cell-based or animal studies, were requested for an app for allergenicity assessment? In this case, the assessment based on additional studies are requested only on a case-by-case basis, and obviously it will depend on the previous information. If there are indications of a specific proteins that might trigger an allergic reaction, in those cases, additional experiments will be requested to the applicant, and this could be in the way of cell-based or animal studies. This is a controversial question because at the moment there are no validated methods, cell-based or animal studies for allergenicity assessment. And this is the reason why a weight of evidence approach is used. And where we are trying to achieve here is to improve or reinforce some of those elements with new methodologies that are science-based and fit for purpose. I give the floor to you, Javier. Thank you.
Thank you, Antonio. Yes, there are other questions. Okay. For instance, the next one is, please mention the names of the new methods which EFSA is exploring that can be used as complementary alternatives to the current method used in protein safety assessment. Okay, just to be clear that we have an open mind with this mandate, so we are trying to look for any potential informative method that is available for research purposes. Antonio already included a nice slide about it, but I mean, just to give you a bit more details, we are particularly interested in looking at bioinformatic approaches based not only on the primary amino acid sequence, but also on the secondary or tertiary three-dimensional structure. For sure, we are also looking at new in vitro cells studies, looking potential toxicology. Also, we are looking alternative in vitro gastrointestinal digestion protocols that could be better mimic the potential gastrointestinal fate of the protein. We are also looking at recent advances on organoids. I mean, we are trying to cover as much as possible the current state of the art of tools, looking at the structural and functional characterization of proteins. Okay, next question is employment of machine learning tools for prediction of possible epitopes being considered as part of in silico approaches? Well, we have indeed received some studies dealing with particular epitopes. At least in the case of allergenicity, one of the problems is in some cases the epitope mapping is so broad and so variable that, I mean, it could give additional information, but not definitive in the considering the current state of knowledge. Okay, there are another question about if I can clarify what is meant by EFSA developmental projects that I included in the last slide, but I think this question is maybe to be addressed by Antonio because he knows much better than me the current ongoing project. So, Antonio, I don't know if you would like to address this question.
Thank you, Javier. Yeah, thank you, Javier. As described in one of the slides that Javier presented, the information that we will consider for developing the scientific opinion will include the survey that we are going to launch these days. It will include the public consultation, and in parallel, EFSA alone is also commissioning work to external contractors, we call procurements, to further deepen into a specific aspects. In this case, we have procurements dealing with allergenicity, to define new strategies and how to perform a ranking of allergens, just to give you some examples. In the case of toxicity, we have a procurement commission to further pursue on in silico tools, as well as what could be coupled with in silico tools from the in vitro perspective. These procurements will come available by the end of next year, beginning of 2025, but most of the information will already be available by the time this opinion will be published and for sure embedded. A couple of more questions from my side. The doses required in toxicity studies should be associated to real exposure instead of those from OECD protocols coming from chemicals. So I guess this question is coming from the angle that several of the requirements in Codex and in the followed by EFSA are based on those borrowed from the chemical risk assessment. And as I mentioned at the beginning, proteins are different. And indeed, this is an element that the EFSA GMO panel will consider and explore on how to make more fit for purpose risk assessments. The next question is, can you define a safe protein mentioned in the slide on the potential to use similarity to safe proteins? In this case, obviously, history of safe use is linked into that direction. It's a concept that intuitively for extreme cases, I would say, is quite easy to understand. We have cases of proteins extracted from maize introduced in soybean, or a protein from oat introduced also in one of the GM plants. And obviously these proteins from oat are known to be safe, are safely consumed. That is what is referred by safe proteins, just some clear example. Would it be naturally occurring protein with documented consumption or previously reviewed protein in other GMO product considered? This is also an interesting aspect that the GMO panel is reflecting on how to use this information on not only on the history of safe use, but also on previous safety assessments of proteins and how that can be extrapolated for new cases. I would say I will pass the floor to you, Javier, if you have any specific question to answer, and then because we are reaching the time. Thank you. Javier, the floor to you.
Okay, yes, maybe I can take just additional questions. Yes, the next one is, what is the smallest acceptable degraded band size after pepsin digestion? Well, to be honest, as far as I know, there is not a consensus. So that's one of the key aspects that we would like to get into detail. For the time being, what I can say is that just if the gel used for the SDS page, depending on the resolution, is just let's say it's the current threshold, which is around 2-3 kilodaltons, but it's not explicit. It's because they can enter into the gel, but there are no, let's say, regulatory or legal threshold about the minimum size. But this is one of the key points that we are working on it. Okay, I think there are just I think we are running out of time. We are almost 5 minutes to finish. So I don't know, Renilde, if you would like to take the floor to wrap up and just to say goodbye to end the webinar.
Yes, I'm afraid there is a couple of questions still in the waiting, but again, some of these questions are very suggestive, from which we can have an indication what the requester has intended, and I'm sure that when with opening the survey, we can still deepen this out. So apologies, but we are running out of time, and I would like to draw this webinar to a close by, first of all, acknowledging everybody who was involved in its preparation. So all the colleagues on slide have been contributing. Thanks you very much for this. And to all of you participants, you can help us further by giving still 5 minutes of your time by filling in the evaluation survey that you will be receiving per mail. It was a real pleasure having you, and again, we were impressed with the high level of interest. We have reached the peak of 257 participants, but for all the others, the recordings are available, as we said, as well as the presentations. Again, any important concern or question can be posted to us via the EFSA platform Ask a Question. Please go there. Also for the procedural aspects and on the authorizations that we have been filtering out for now, you can post them there and you will get an answer. Scientifically, I think an important take-home message is that genetic modification in GM plants over the last 20 years can be used in a safe way. We have assessed many cases, and our experiences with the safety testing of the newly expressed proteins and the GM plant itself, they have learned that the general safety of these products for consumers, animals, and environment can be is in effect. On a case by case, you can actually read our opinions to see if and where we would have identified a specific hazard or a very circumstantial risk. And these we cannot wrap up, but each opinion will be informing on that specific GM product. Now, with more and more innovative products and new traits, again, a practical modernization of protein safety is needed, in our view, in order to respect also the highest level of consumer protection while we are being introduced with new proteins, new way of nutrition, and we want to respect that highest level, but also respect the reduction of laboratory animal sacrifices. So that's where we have to go in the future and strike the balance right. So new approaches are needed, and we ask all stakeholders to collaborate. We will do that also by probing in the international context with organizations that are developing methodologies and are concerned with new methodology approaches. We have a plan in place for such consultations. As already mentioned, we are opening now the survey to collect further inputs, and that survey will be open until the 18th of February. We expect contributions with scientific detail, eventually experiments or products that you are assessing, but any of the unanswered questions or suggestions may also be further elaborated in there. A last message is to please subscribe to the newsletter of EFSA, because very regularly we have stakeholders initiatives, not only like this, but also on other topics. And through that channel, you will also be made aware of the public consultation on the text of our draft opinion once it has been developed by the GMO panel. This is expected by the second half of 2024, and it will be online for commenting for 2 months. So you will have another occasion to interact with us and give us the scientific detail to deliver a robust opinion. We look forward to seeing all of those scientific contributions and your ideas for protein safety assessment now and in the future. And with this, I really hope you enjoyed the seminar, and I close it wishing you a very good day. Bye-bye, everybody. Thanks a lot.